Thank you all for coming this morning, and it's my pleasure to introduce today's Grand Round speaker, MWendt, who's visiting us from Purdue University. Mike received his PhD training at just next door at the Medical College of Wisconsin, where he worked on chemokines and in the gut. He then moved on to do his postdoc training at the University of Colorado in Bill Schumann's lab, where he continued his interest in complicated signaling pathways in cancer by working on TGF beta and EMT. He then moved with Dr. Schumann to Case Western, where he continued his postdoctoral training and then started his faculty position at Purdue. I met Mike a couple of years ago, or a couple meetings ago, through a mutual friend, and was very excited to find out that he worked on FGF receptor signaling in breast cancer, which is my favorite pathway of interest as well. And we were sort of commiserating last night about FGF receptors in breast cancer. That's been known for a long time. They're very important, but it's a very understudied signaling pathway. There are actually very few labs in the country that work on FGF receptor signaling. So I was super excited to meet him and thought it would be fun to have him out and hear what he's doing in his lab now on growth factor receptor signaling and targeting them in metastatic breast cancer. So thank you for coming out today.
I always get nervous at the pre-talk applause. I really got to deliver. Well, thank you, Kaylee, for the invitation. It's a pleasure to be here for the seminar and the meeting later this week, so it should be a good couple of days. So today I just want to talk to you briefly about some of the recent work we have going on in my lab, since a little bit of my postdoc work, and then some of the new stuff we've been able to accomplish over the last few years at Purdue. It's really centered around this idea of how growth factors can change between a primary tumor and then the metastatic tumor. Right? So if you think about pharmacology, we're really trying to target these metastatic tumors, but I think work going on from our lab and other labs really suggests that the metastatic tumor can change pretty drastically as compared to the primary tumor from which it was evolved. And so if we look a little bit closer at the process of metastasis, it's really this kind of sequential cascade of events. And so we have formation of the primary tumor here in the context of a normal epithelium. And then we think a lot about this process of EMT. And so these primary tumor cells will shed those epithelial characteristics. And a lot of work has gone into characterizing how that process of EMT facilitates the invasion and dissemination of those tumor cells. Right? But then, metastasis has to carry on several more steps to eventually form a productive secondary tumor that's going to impede the function of a vital organ. And so EMT is probably more accurately described as this process of plasticity, or EMP. Because if we look at what eventually forms here, clinical biopsies and several different models, you really can see that the eventual metastasis takes on an epithelial phenotype that can be very characteristic of the primary tumor from which it was derived. And so this kind of changing back and forth between a mesenchymal characteristic and an epithelial characteristic is kind of really at the heart of what we're interested in studying. And so breast cancer remains a lethal disease. So last year, 250,000 diagnoses, resulting in nearly 40,000 breast cancer lethalities. And if we look at these stats a little bit closer, it's indeed the women that are walking into the clinic being diagnosed as having disseminated disease. And so a lot of these processes that we're interested in biologically have already taken place. Cells have already evaded out of the primary tumor. They've already survived in the bloodstream. They've already initiated growth at that secondary site. And so, again, if we think about targeting late-stage metastatic disease, we really need to understand more about what's unique about the growth of those metastatic tumors as compared to that primary tumor. And so we get back to this idea of EMT. EMT is not solely a process that's associated with metastasis. It's indeed a physiological process. And if we think about wound healing, so a lot of my work in graduate school was looking at wound healing in the gut, or the skin, for instance. You have an intact epithelial monolayer that will get wounded. Those cells along that wound edge will migrate into that denuded area and undergo an EMT as they do so. But then they're going to reacquire epithelial characteristics to reestablish that intact monolayer. And that's probably a very conserved process. because these cells are remaining in that same physiological location. But if you think about other types of EMT that go on during development, or pathologic EMT that's associated with metastasis, once these cells undergo a mesenchymal transition, they're then going to leave that physiologic location and migrate to some other place. So if you think about development, these cells are migrating out of the neural crests, and they're going to go on to develop into mammary epithelium or gut epithelium, which obviously is very different from that original cell that they're derived from. And so we think about pathologic or metastatic EMT along those same kinds of lines. A mammary epithelial cell that undergoes a transformation event is going to undergo EMT and then migrate to some metastatic destination, the bones or the lungs, central nervous system. And obviously the microenvironmental influences in that secondary location are much different than that primary tumor. So a really driving hypothesis is that differences in what we characterize as this primary epithelial state and this secondary epithelial state may be at the heart of why metastatic tumors are failing to respond to a lot of the therapies that we're using based on what we know about that primary tumor. And so just a good example of this process of plasticity is this model, which we'll talk about a few times today, is the 4T1 model. So the 4T1s, if you're not familiar with them, are a highly metastatic model of breast cancer. So here we inject 10,000 cells onto the mammary fat pattern within a period of 5 to 6 weeks. Those mice go on to get a very large primary tumor that will progress to multi-organ metastasis. And so here, we've taken those cells with a firefly luciferase that's driven by the E-cadherin promoter. So E-cadherin is kind of a prototypical marker of epithelial cells that goes down during EMT, both at the protein level and its transcription gets inhibited. And so here, if we just look at some pictures, we can see a drastic EMT that takes place in the primary tumor, and then in these early mets, those cells are still in a mesenchymal state. We see very little firefly signal, and by IHC of that same tumor, we see very little E-cadherin expression. But if we look at these later stage tumors, about a week later, where these lungs are completely encapsulated with tumor tissue, we start to see a signal from the firefly come back, and by IHC we can start to see E-cad again. And the beauty of this system is that these cells are stably expressing firefly under the E-cad promoter, but they also express ranilla luciferase under a constitutively active promoter, and so we can essentially do an in vivo reporter assay. So again, if we take these cells, we grow the 4T1s on plastic, they're very epithelial. They look epithelial, they express a lot of E-cadherin protein, and here the firefly reporter is very active. We can suppress that by treating these cells with TGF-beta, common way to induce EMT in several different cell types. But if we look now, comparing cells growing on plastic to those of the primary tumor, we can really see this massive EMT that takes place, this spontaneous EMT that takes place when these cells are injected into a mouse. Importantly, however, if we compare these to these late-stage metastases shown here, we can start to see that MET reaction that's taking place. And so just a really good example of a cell line that on plastic looks completely different than what it does in vivo, and we can really see this ability of these cells to transit between an epithelial state and a mesenchymal state. Again, consistent with the fact that they're highly metastatic. And so we think about this plasticity. There's a nice review last year from Max Wisch's lab that did a really good job of matching up this idea of epithelial cells, mesenchymal cells, and then these plastic cells. They call them these different types of stem cells, but we'll just refer to them as these plastic cells for today. And they did a good job of matching those up to the breast cancer subtypes. Well, that breast cancer is not a single disease. It can be categorized molecularly into several different subtypes. Here they show 5 different subtypes. And again, these are all based on primary tumors. And so several recent studies have shown that if you take the PAM50s, a gene set of 50 genes that can boil down to subtyping these breast cancers into these 5 different subtypes. If you do that, comparing primary tumors to metastatic tumors, indeed, several primary tumors have been shown to essentially change their intrinsic subtype based on the metastatic process. And so could this idea of switching between epithelial and mesenchymal phenotypes be at the heart of these changes in phenotypes? And so if we look at kind of a flow plot like this, I like these plots. A lot of people use them to look at, represent changes in polymorphism. But if we think about it in the same way in terms of an epithelial mesenchymal makeup, if this is the makeup of the primary tumor again, you're going to have, and this is where you're getting biopsy material from. You're going to have surgery and therapy and metastasis, resistance to that therapy that's taking place over a period of years. And so again, the eventual metastasis that's going to lead to lethality in the patient is probably going to be much different than the primary tumor. And so really, could this idea of plasticity be at the heart of these changes or discordance in these important therapeutic targets that we're trying to inhibit? And so to do that, we looked at an example that in breast cancer that really seemed to represent this problem. And so inside that basal-like subtype of breast cancer, there's a clinical diagnosis of triple negatives, which is a very uninformative diagnosis of patients lacking HER2 amplification and lacking ER and PR. But if we look at those patients and categorize them to those that have high levels of EGFR versus low levels of EGFR, epidermal growth factor receptor, those patients that have high levels of EGFR definitely have decreased survival rates. And so these data, so this is just a representation of the triple negative phenotype where they don't have ER, so they're not candidates for hormone therapies. They don't have HER2, so they're not candidates for a lot of these new HER2 therapies. And so that these data led to this idea of targeting EGFR in the triple negative context. And so through the 2000s, early 2000s, there were several clinical trials testing erlotinib and gefitinib for treatment of metastatic triple negative breast cancer. And by and large, these trials were not very successful. It didn't seem that specific inhibition of EGFR led to any kind of clinical benefit for these patients. And so that really kind of posed this question for us. Why is there this disconnect between the prognostic value of EGFR and its inability to be a good therapeutic target? So along with that, there's a lot of good basic science research from Jeff Wyckoff and John Connolly showing that EGFR participated in this idea of primary tumor cells evading out of the primary tumor and following this gradient of EGF that's put down by macrophages, tumor-infiltrating macrophages. So we started looking at that in terms of EMT. We're able to show that indeed those cells that have been pre-treated with TGF-beta to undergo an EMT are at an increased potential to follow this EGF gradient. And so again, now you have clinical data, basic science data supporting this idea of EGF driving systemic dissemination. But again, why not being successful as a late-stage clinical target. So through these studies, we created this model in which we took normal murine mammary gland cells and immune G cells, which it's hard to say at first, but after a while it rolls right up. And we were able to show with those cells that if you overexpress EGFR, you're able to essentially transform those cells. So here's polyomyl-T, the well-established molecule that will transform normal mammary epithelial cells. And it's similar to that, if we overexpress EGFR, we get tumor formation compared to control cells. And so this was an interesting finding, but what really came out of that was the idea that the tumors that form are a good model of in situ carcinoma. So here over 5 weeks, we get nice tumor formation, but we can cut those tumors off and follow those mice for several weeks after that and really don't see tumor recurrence or metastasis. However, if we take those cells and treat them with TGF-beta again to drive EMT prior to tumor cell engraftment, so here we have upregulation of fibronectin, loss of ECAD, some of these classic EMT markers. The tumor that eventually forms is a little bit bigger, but more importantly, after surgery, 100% of these mice will get tumor recurrence and kind of this broad spectrum metastasis. So we just look at some histology. We see nice ECAD here and expression here and very kind of ductal formation. But in the post-EMT tumor, we have much more invasive looking tumor. And we can really see this kind of broad spectrum metastasis that takes place in these mice.
Can I ask you a quick
question
then about those primary tumors between the pre-EMT and the post-EMT? If you're just looking at it morphologically, so not at markers, do you see different morphology? Do you see different invasion? Do you see a different pattern of expansion, kind of like essentially a cohesive front versus single cell infiltration?
Right, yeah, definitely. So maybe this histology doesn't portray that the best, but essentially we see all those kind of classic characteristics. Where here in the pre-EMT cells, it's definitely a very ductal tumor. Where in the post-EMT tumors, you definitely have a very nice invasive front. You can see single cell invasion into the fat pad. Things like that, for sure. So whatever's happening during that pre-treatment here, there's definitely kicks off some kind of signaling pathways in vivo to carry that process through, which I think we'll get back to, that doesn't take place in these cells in in vitro. So we see that a lot with TGF-beta, where it can be a feed-forward process in certain systems, but not always. So then if we look just at pulmonary metastasis, we can see that, again, the pre-EMT cells after surgery, we really don't see pulmonary metastasis, but the post-EMT tumors go on to form pulmonary metastases. And so we're able to subculture these cells, and we call them the enemy lung metastatic cells. And if we take those cells and put them back onto fat pads of a new group of mice, we get much more robust primary tumor formation. We do the surgery, and those tumors come right back again. So that allows us to subculture out those cells and form this progression series. So now we have the NMUMG cells that are not transformed, normal mammary epithelial cells, the transformation by overexpression of EGFR, and then these two progressively metastatic cell lines. And if we look at these, they've definitely undergone a MET. So we can see morphologically they are a little different, but by classic kind of EMT markers, ECAD is back, Momentin is down. These cells have reverted back to an phenotype. And so this kind of now allows us to ask the question of what is this impact of EMT and metastasis on EGFR signaling?
I'm sorry, maybe you mentioned this, but are the original cells, are they, oh my gosh, what's the question here? Are they immortalized?
Definitely.
So
Yeah, the NMUMGs, quote, normal, right? So they're very immortalized. They're probably right kind of on the border of forming tumors or not.
And so then the EGFR kicks them?
Yeah. Yeah, so the overexpression of EGFR is probably not a good... Overexpression of EGFR is not going to transform like primary. So that's an interesting why that is. I'm not quite sure in this model. So in doing this, did some actually control experiments very early on, showing that we can treat mice with erlotinib, a very well-established EGFR inhibitor in lung cancer, and very nicely prevent tumor formation. So just consistent with the fact that this is a transformation model that's driven by EGFR activity. However, if we take those LM cells, the metastatic cells, tumor formation is delayed, but upon resection we see really no difference in tumor size between the no treatment group and the erlotinib group. And then if we look at tumor recurrence after surgery, the erlotinib group is actually a little bit worse than the non-treated group. If we look earlier on, so these are bioluminescent measurements that take place shortly after primary tumor engraftment. So we can see this ability of EGFR to drive primary tumor dissemination, right? So here in the LM group, if we're comparing no-stim versus erlotinib, we do see a significant decrease in the cells that are disseminating. So again, going along with this idea of EGFR playing a role in primary tumor dissemination. But at the end of the day, all of these mice in the LM group go on to get metastasis, and we see really no difference in survival. And so taking these cells, we asked some very basic questions. First of all, we saw an interesting decrease in EGFR expression. So again, here the enemies were artificially overexpressing EGFR here, so they have high levels of EGFR. But now the metastatic cells are actually down-regulating that driver gene. And if we take those cells and treat them with an EGFR inhibitor, AG1478, we can get an apoptosis by clean PARP. And we see the exact opposite effect in the metastatic cells. So here if we treat with EGF and activate the receptor, we see very robust induction of apoptosis. And if we do a 3D culture assay, so here we take cells, we lay down a bed of a matrix, very similar to like a matrigel, and then place the cells on top of that, and they form these three-dimensional structures, and the growth of which we can track by bioluminescence. And so if we do that, we see pretty nice growth with the NMEs, and that can be inhibited by the EGFR inhibitor, the AG1478, and can be driven by addition of EGF into the media. And in fact, we see the opposite result when we add EGF into the media of the metastatic cells. So again, consistent with the idea of apoptosis induction, EGF is definitely inhibiting the 3D growth of these metastatic cells. Again, the opposite result of what we see with the primary tumor cells. And so that came to Purdue and started acquiring graduate students. And so my first graduate student, I posed this question to her very simply, just let's look at some downstream signaling pathways of EGFR and see what may be at the heart of this change in function of EGF. And so Rima did a really good job of first of all making a much better schematic than I was ever to make about this series. So again, we have the NMUMGs transformed by EGFR, passage through a mouse, harvested from the lungs to get the LMs. passage through the mouse again and harvested from the lungs to get the LM2. And so if we take those cells and just treat them with EGF, we see nice activation of ERK even in the control cells. So here we have the endogenous EGFR levels, and then we overexpress EGFR to transform, and then again we see this drop in EGFR levels in the metastatic cells. If we treat those cells with EGF, even though they have much lower levels of EGFR, almost consistent with the control non-transformed cells, we're seeing this very robust activation of STAT1. So again, a very fundamental change in the downstream signaling pathways that are taking place in these metastatic cells compared now to normal cells. We just do a nice dose response and really show that difference in response. So here again we have high levels of EGFR but barely getting STAT1 activation, whereas these LM cells have much lower levels of EGFR, but they're definitely hooked up to this STAT1 pathway. So STAT1 had previously been linked to induction of apoptosis by EGF. And so Rima did some good control experiments, just blocking EGFR with gefitinib. And then what she found was, however, if she included trametinib, so trametinib is a MEK inhibitor, which blocks the MEK-ERK pathway. When she did that, she very nicely blocks ERK, but she saw this increase in the ability of EGF to activate STAT1. So this is in the NME model. So we get this minimal activation of STAT1 that can be now driven if we kind of shift the balance by blocking the ERK pathway. And so in fact, if we look at apoptosis, again we see this induction of apoptosis with EGF in the metastatic cells, but now if we include trametinib in combination with EGF, we see this dramatic increase. So again, by driving EGF signaling to the STAT1 pathway, possibly the STAT1 pathway, we see this increase in apoptosis. And so now she took it back to the NME cells. So these are the primary tumor cells. Again, they're proliferative in response to EGF. And so here with EGF inclusion, we see a slight decrease in caspase activity. Trametinib here is at a very low concentration. So we're not seeing any apoptosis with just trametinib alone. But now even in these originally proliferative cells, if we modulate those downstream signaling pathways, we can start to see induction of apoptosis by EGF. We can really see how the cells start to round up in a typical apoptotic morphology. She's done a nice job of expanding this to a lot of different cell lines. So this is some other EGFR expressing cell lines for the 468s. Again, the 468s had previously been shown to induce apoptosis in response to really super physiologic levels of EGF, but here Riva's using much lower levels of trametinib and EGF. So alone, not really seeing a response, but then again, in combination, increased STAT1 and apoptosis. The BT20s are a similar cell line that express EGFR. We get a STAT1 response here, but then again, with the inclusion of trametinib, we see that. So Reed was very excited about moving forward into some in vivo experiments, possibly combining this idea of actually EGFR agonism in combination with trametinib. But metastatic cells are smart, right? And so to get around this, we observed a couple of different things. One of which is what is kind of happening in our artificial model, is that metastatic cells tend to downregulate EGFR. So here's the 10A model, and these cells are driven by RAS, and then similarly passage through a mouse to get progressively more metastatic characteristics. And so here the CA1A cells are the metastatic cells. We can see that they similarly lose EGFR. The 231s are a good example of a cell line you can put onto a mouse, and you'll get a couple of METs, and then you can harvest those METs and select for more metastatic cells. If we do that using some lines that we've developed that go to the lymph node or the lungs, we similarly see loss of EGFR compared to the parental cells. But not all tumors are indeed losing EGFR, and so another mechanism that we've observed that metastatic cells might regulate their EGFR signaling is upregulation of this negative regulator of EGFR signaling called MYG6. So MYG6 is a protein that binds to the cytoplasmic tail of EGFR and essentially inhibits all the downstream functions of that receptor. So if we just look at the TCGA and graph EGFR expression to MYG6, we see a slight positive correlation. And it's not very robust, but again, if you would expect EGFR to be a driver, then you would expect a negative regulator of that molecule to be a negative regulation, or negative correlation, as opposed to this positive correlation. And if we look at the CCLE database for cell line correlation, we see a more robust correlation between these things. So again, the 468s express very high levels of EGFR, slightly lower levels of MYC6, consistent with the fact that you can drive an apoptosis in those cells. So we took the 231s, which are kind of middle-of-the-road levels of EGFR, but very high levels of MYC6, and knocked down MYC6 in those cells. And the outcome, essentially, is that we can potently inhibit the ability of these cells to undergo pulmonary tumor growth. So here we're just doing a tail vein assay. We inject cells into the tail vein, get very nice pulmonary tumor load right after injection. And 2 days later, you tend to see a massive reduction in those cells. A lot of those cells will undergo apoptosis. But with MYC6 knockdown, we see an even greater death of those cells, and they're never really able to recover from that. And so again, this idea of MYC6 regulating EGFR activity and preventing apoptosis in these metastatic cells. So, just to summarize this first part, EGFR is a potent driver of primary breast cancer. It definitely transform a model of normal epithelium. EMT and MET enhances this linkage between EGFR signaling and STAT1, and forcing this change in signaling by using trametinib in combination with EGF, we can essentially mimic this change in signaling and get an apoptotic response to EGF. And so, given that many metastatic cell lines seem to upregulate MYC6 or downregulate EGFR, we're currently exploring some different ideas of how we might be able to activate STAT1 in terms of interferons or cyclophosphamide in combination with trametinib as a potential therapy. And so in the second part, I kind of want to talk about what's taking the place of EGFR, right? So if EGFR is being downregulated or inhibited or functioning in a pro-apoptotic function, what's driving the growth of those metastatic cells? And so to do that, we went back to our EGFR expression model and did a very simple experiment where we treated the cells with TGF-beta for 4 weeks, and that drives a very robust EMT. So here we're replacing the TGF-beta every few days, and we can see that these cells lose all kind of epithelial characteristic and really turn into a very kind of fibroblast-looking cell. But it's been known for a long time, if you simply take the TGF-beta away from those cells, they'll revert back to an epithelial phenotype. And so you can just see that. We have MCAD coming up over the period of 4 weeks, and then it goes back down as we withdraw that exogenous TGF-beta. So this goes back to this idea that in vitro, these cells are not able to feed forward that mechanism, whereas they seem to be able to in vivo. But importantly, what we see is this progressive loss of EGFR. So we're able to recapitulate what we were noticing in vivo simply by doing this EMT and MET protocol. So we took these two cells, again, what we would call the primary epithelial state and the secondary epithelial state, and ran a microarray to characterize those states. And so in addition to loss of the receptor, we see downregulation of several EGFR ligands, but more importantly, we see upregulation of FGFRs. So here we have FGFR 1, 2, and 3, all being upregulated in this secondary epithelial state. And so maybe as Kaylee has told you guys, FGFR is an awesome receptor. It has these 3 extracellular IG domains, and it undergoes a lot of differential splicing, which really increases the repertoire of receptors that can be expressed by different cells, right? And so there's 23 different FGF ligands, so in combination with different receptors and different ligands, you really can get this plethora of different signaling pathways that take place. And so we'll simplify it all down to looking at 2 isoforms, the 3B and 3C. So the 3C isoform is known to come up during an EMT. So FGFR definitely gets transcriptionally driven during an EMT, but then there's splicing factors that get lost during an EMT, and that allows for this upregulation of the specific 3C isoform. And so we see that, but more importantly, if we look at the metastatic cells, they have constitutive expression of FGFR now. So again, this idea of FGFR coming up during EMT, but doesn't go back down. Unlike ECAD, for instance, which goes down but then comes back. And so we also see it that's in this beta isoform. And so the beta isoform has been linked to more of an oncogenic role of FGFR as well, as it lacks this outermost IG domain. And so Wells Brown, my first postdoc, came into the lab and did some really simple experiments to start with. He did a lot of immunofluorescence as a graduate student, so we started with that. And so we can just see in the YFP and MUMG cells, we have nice expression of ECAD, very junctional. Actin is similar, and then if we treat these cells with TGF-beta, we lose ECAD. It's probably isn't the best here, but the actin turns into very filamentous actin, very classic EMT phenotype. And then he was also able to establish a twist overexpressing in an MUMG model. So twist is kind of a prototypical transcription factor known to drive EMT in several models. So here in the MWendt model, we have a very nice EMT that happens with twist expression. But more importantly, when he takes these cells and stimulates with FGF, we can see that in contrast to EGF, which seems to be about the same in terms of an ERK1/2 activation, FGF2 is only able to activate or phosphorylate ERK1/2 following induction of EMT, in this case by TGF-beta and also by twist. So here we have YFP expressing cells or twist expressing cells, and we stimulate with FGF2, and only the twist expressing cells are responding to the FGF2. And here we can just see the nice EMT markers that take place during this twist-induced EMT. Again, loss of ECAD, gain of NCAD, and here we can see gain of FGFR. And if we look a little bit deeper about where the protein is being expressed in these cells, right? We can't see it in control NMUMG cells, but in the twist overexpressing cells, Wells does some immunofluorescence, really see it kind of throughout the cell. See some in the nucleus, see some in the plasma membrane, throughout the cytoplasm. And this was kind of in contrast to what we were noticing in vivo. So if we look at the protein atlas, if this is a patient, a primary tumor, a ductal carcinoma, and we can see that FGFR is very junctional. Here's a lymph node metastasis, where we kind of see something more like what we were seeing in vitro, with FGFR expression being kind of all over the cell. This is consistent with differences in ECAD-adherent expression. So here these ductal carcinomas are very ECAD-adherent positive, but these the lymph node metastases are ECAD-adherent low. And so to look at the potential role of EMT in regulating this differential localization of FGFR, we overexpress the GFP-tagged FGFR1. And so just like the ductal carcinoma, if we do that in NMUMG cells, we definitely see very junctional localization of FGFR1. However, if we take those same cells and then just treat them with TGF-beta, those junctions break down, and now FGFR becomes much more looking like what we see in the twist-expressing cells. So FGFR1 is kind of getting this idea of EMT not only upregulates expression of FGFR1, but has an impact on where the receptor is located throughout the cell. And so there's a lot of literature in the late 90s, early 2000s about the role of FGFR in its interaction with different integrin proteins, particularly beta-3 integrin. This was primarily in endothelial cells, where they showed beta-3 can interact with FGFR, beta-3 can bind FGF2 and essentially act as a co-receptor for FGFR. And so it came out of Bill Sheeman's lab. If you don't know, beta-3 kind of drives the universe in Bill Sheeman's lab. So I know that, I knew that beta-3 expression had a very nice morphological impact on the NMUMGs. And so we simply overexpress beta-3 in the context of our FGFR-GFP construct, and we see a similar dislocalization from the junction. And this was very interesting because beta-3 expression doesn't drive a full-on EMT in these cells. It's really just a morphological change. So here, ECAD's not going down. But just by overexpressing beta-3, that was sufficient to get FGFR out of these junctional complexes. And here we can see that by co-IP. So here if we pull down for the GFP tag on the FGFR, we see very nice interaction with ECAD-adherent, that junctional protein. But again, if we overexpress beta-3, we can prevent that. And we start to see a little bit of interaction with beta-3, but probably not to a great extent, given that both of these proteins are overexpressed. What's more important is that if we look at downstream signaling, so here we have control cells, we see a little bit of a response to FGF2. If we overexpress beta-3 alone, that doesn't really change. More importantly, if we overexpress FGFR alone, that doesn't really change, right? So now when FGFR is in this junctional complex, it doesn't seem to be functional. But again, when we overexpress both of them and move into this more cellular diffuse localization, we see very nice response to ligand. And so again, trying to get away from these overexpression systems, we sought to utilize this D2A1 model. So the D2A1s, similar to the 4T1s, they're a mouse model. You can put them back into a fully immunocompetent BALC mouse. And they're very nice because they're paired with this OR model, which is a model of dormancy versus metastasis. And so here, a tail vein experiment can show that the ORs will go in, and a lot of those cells will die, and they'll remain dormant over a period of several weeks. Whereas the A1s are a typical metastatic cell line, and they undergo outgrowth. The A1s happen to lack EGFR compared to the ORs, consistent with what I was telling you earlier, this loss of EGFR in more metastatic cells. But getting back to this idea of beta-3, we can see if we take the A1s, we get a response to FGF2. If we knock down beta-1, we've known for a long time, when you knock down beta-1 integrin, you have this compensatory upregulation of beta-3, and that's consistent with a better response to FGF2. But more importantly, when we knock down beta-3, we almost completely prevent the ability of those cells to respond to FGF2. And here, again, if we just use a 3D culture approach and track the growth of those cells by bioluminescence, we can see FGF2 can drive the growth of the A1s, but more importantly, when we knock down beta-1, there's this impact on basal outgrowth of those cells. This has been shown by several groups that beta-1 is important in this model as well as others in terms of growth in 3D culture and metastasis in general. But now if we include FGF2 in that culture, we can completely rescue that phenotype. But again, with beta-3, we see a slight impact on basal growth, but completely nullify the ability of FGF to act as a protective factor in these cells. And so here, if we knock down FGFR in the A1s and do that same tail vein injection, we can very nicely prevent pulmonary tumor formation using 2 different shRNAs. And we see this impact on ERK1/2 signaling. And moving to some pharmacological means to target this system, we started to use BGJ398 as a compound from Novartis, which is a very nice specific FGFR inhibitor as opposed to some other molecules that are a little more pleiotropic. PD-173074 is a lab drug that's been used for a long time. It's also very specific, but not suitable for in vivo stuff. But importantly, if we use a FAK inhibitor, so downstream of integrins is the focal adhesion complex, right? A major player there is focal adhesion kinase. And so there's some nice inhibitors to FAK, and so PF271 is a compound from, used to be from Pfizer, and now it's bought by another company, I can't remember. But if we use that, we can very nicely block the ability of FGF2 to induce ERK1/2 phosphorylation. So we use either of those drugs in vivo. We can, again, do a tail vein injection, let those cells set up for 7 days, and then initiate treatment, and very nicely delay the ability of those cells to undergo pulmonary growth. And so about this time, we started a collaboration with Nathaniel Gray, who is a synthetic chemist, who was making covalent inhibitors to FGFR. So BGJ is a competitive inhibitor to FGFR, very similar to like erlotinib or most of the clinically approved kinase inhibitors. They're in constant competition with the cellular pool of ATP, right? And that can really limit their efficacy. And so there's a resurgence in this idea of making your kinase inhibitor in such a fashion that it forms a covalent bond with a cysteine residue that's in or around that ATP binding pocket. And so we can see, I don't think you have to be an organic chemist to appreciate that these scaffolds are based much upon the BGJ scaffold, but then have these adducts here that are able to form a covalent bond with cysteine 474 or 477, which is right by the ATP binding pocket. So if we use these, again, in this ERK assay in the A1s, here we pre-treat the cells for a long period of time with the drugs before we stimulate with FGF2. And so here we can get blockade with BGJ, but much better with the covalent compounds. And again, using the 3D culture assay, we can really show the improved efficacy of these compounds compared to a competitive inhibitor. So here, cells were grown up for several days in 3D culture and then just exposed to the drug for a short period of time, was washed out, and then these readings come several days later. And we can see that even the short exposure to drug with the covalent compounds leads to this long-term inhibition of cell growth as opposed to the competitive inhibitor. And so, I hesitated to put this in because I'm not a cholesterographer at all, but we can really see the compound here, right? So here are those rings that are right here. And then it bends around and forms this covalent bond with cysteine 477. And that pulls down on this P loop and really drives FGFR into an inactive conformation, which is significantly different than what's happening with a competitive inhibitor. This idea that the covalent binding actually changes the molecule into an inactive conformation, much like a type 2 kinase inhibitor. And so I won't belabor the pharmacology, but suffice to say FIN4 was much better than FIN2, the other covalent inhibitor, with pretty good in vivo pharmacology, but very good IC50. So here we're down to nanomolar concentrations for IC50s, and pretty good specificity for the FGFRs. And so, just to finish up, we've done several in vivo experiments now characterizing this FIN4 molecule. So here, if we go back to that 4T1 model, where we have primary tumor formation, that where we can do surgery, and then those go on to metastasize. If we do that, and then take those pulmonary metastases and simply do a single cell suspension, but don't do any kind of selection. So here we're taking that whole microenvironment, essentially, immune cells, epithelial cells, endothelial cells, and just place those into our 3D culture model. We can very nicely see that through the metastatic process, again, the 4T1s, when you grow them on plastic here, these in vitro cells, they're very epithelial. They don't have very much beta-3, they don't have very much FGFR1. But again, the metastatic cells upregulate these markers. But more importantly, if we take those heterogeneous cultures and treat them with FIN4, even at 60 nanomolar, we can almost completely block this kind of constitutive phospho-ERK that's going on. So whatever's driving this basal phospho-ERK in this heterogeneous ex vivo culture is very dependent on FGFR signaling. And so if we take those again and put them in 3D culture for 3 days and let those structures form up, we can add FIN2 or FIN4 at that point, and as my postdoc likes to say, we can crush those. And so that's very, very encouraging. And so if we do the in vivo experiment here, so again, we let the metastasis form. We can take those pulmonary metastases, instead of putting them in a 3D culture, we simply put them back in the tail vein of a new group of mice to kind of normalize the field of pulmonary load. If we do that, let those set up for a day or two, and then start treatment. And here we're only going at 25 mg per kg every 48 hours. So not a very heavy dose of the kinase inhibitor at all. And we can really nicely prevent pulmonary tumor growth. And so these are mouse models. If we look at some human data, we can show that FGFR1 alone, or FAC alone, so this is the gene name for FAC, really doesn't bifurcate patient populations in terms of survival. Beta-3 does a nice job, but if we put them all together, we see this really nice difference in survival based on patients that express low levels of these three genes versus patients that express higher levels. So now we've started to move into these PDX models a lot more. So if we take a patient-derived xenograft from a brain metastasis, from a triple-negative breast cancer patient that progressed on chemotherapy, take these directly from the patient. These PDX are passaged strictly in vivo. They won't grow in a plastic dish. And so if we do that and expand them up to a couple of cohorts of mice and put them on there, and these cells have a really long latency, so they'll stay for about 42 days before we see pretty consistent outgrowth. And so if we do that and let those set up for those 40 days and then start treating it again, the 25 mg per kg every other day, we can very nicely prevent growth of these PDX tumors. So a little bit of time left, so good. So getting back to this idea of EMT and MET driving this discordance or lack of expression of therapeutic markers in the metastatic setting. And so we talked about EGFR in this idea of basal carcinoma or triple negatives, but what about HER2? So HER2 is a large subtype of breast cancer, and it's been shown pretty consistently that if you look at HER2 expression between primary tumors and metastatic tumors, there are definitely a set of patients that show this discordance in HER2 expression. And those patients that show discordance have a much decreased survival rate. As you imagine, they're not responding to those HER2 target therapies anymore. And so a lot of work, which I don't have time to talk about today, we've really gotten into this idea of EMT and MET and driving discordance of HER2. And you really can see it here. So if we take an HER2 transformed cell line, we can easily get resistant populations to a kinase inhibitor for HER2 and EGFR, patented, which is a clinically used drug. We can also drive a resistant population by TGF-beta. But more importantly, if we take those cells and treat them with this new therapy called TDM1, which is trastuzumab, which is a therapeutic antibody for HER2, that's tagged with these microtubule inhibitors, a very potent cytotoxic drug. If we do that, only the cells that have undergone a TGF-beta-induced EMT and recovery are able to now downregulate HER2 and take on this discordant phenotype and become resistant to this TDM1. And this seems to, again, be important clinically. Just give you the punchline here. If we get down to a PDX model here, so this patient progressed on trastuzumab and chemotherapy. And so if we take these PDX tumors and grow them up we can show that at concentrations of TDM1 that completely block tumor formation in a sensitive model, these PDX are completely resistant to TDM1. However, if we take those tumors off, or take those mice off that therapy, move them to FIN4, we can very nicely prevent the outgrowth. And so here we can see that these tumors really don't express that high level of HER2 anymore compared to a HER2 over-expressing cell line, but we can detect FGFR protein. And so I'll just stop there by saying that EMT MET cycles can severely alter or fundamentally change drivers of growth between primary tumors and metastatic tumors. I showed you data to suggest that EGFR STAT signaling seems to be increased following an EMT and metastasis, leading to a negative selection against EGFR expression. And the processes of EMT really work together to drive FGFR signaling. And finally, FIN4 seems to be a good candidate for further drug studies in terms of inhibiting FGFR in the metastatic breast cancer setting. Right, so if these primary tumor events are HER2 driven, possibly these later tumor events might shift to an FGFR mechanism. And so with that, I'll show a nice picture from my backyard, and just thank Wells, who is doing the FGFR story and that HER2 story, and then Rima, who's really doing a nice job characterizing that change in FGFR signaling through metastasis. I mentioned Nathaniel as a collaborator. Elena Whelm was key in hooking us up with some of the PDX tissues and getting us going in that direction. Joe Davison is someone in the department who we collaborate with a lot, and Kathy Carlin is the person at Case Western who really got me into EGFR signaling and those things, and funding from these sources. So with that, I'll take any questions.
What's the toxicity of your FIN4 in those mice? I
Yeah.
assume that covalent bonding to other cell types might not be
Right.
a good idea.
Right, so like the FGFR antibody has been very kind of toxic. So at this dose, which we've kind of started low for real life reasons because we use less drug, but because it's it's pricey. But yeah, so if we treat every day with the 25 mg per kg, we definitely start to see mice lose weight. At this dose, we've treated 4 weeks. We really don't see any change in terms of and we actually see the increase because a lot of these mice are starting to get sick over a while. So these are good experiments that we should probably do more as we get more into this in terms of long-term treatments and see if we do see some of these toxicities. Because yeah, I mean definitely FGFR is physiologically important.
Is there a reason that when you were treating the mice that, okay, so you have the mice that have the lung mets, I think it was, I can't remember if it was 14 or something, and then you went in to treat with a FIN4 inhibitor?
Yeah.
Instead of treating the actual mets, you took the mets out and then put them in with the
Because even the 14-1 model, your variation there, and at that step, you can have some mice that have mets and some that don't. So it's kind of tough. And these are definitely experiments we want to do, like trying to establish a window. Can we regress those mets?
Exactly.
Yeah. So for these, this is kind of the first go at it. So in this case, we wanted to just take those out, kind of try to maintain that microenvironment as best we could, and just kind of normalize the field. So we can do a tail vein injection there and know that every mouse, it's just much more consistent. So really just kind of to get a feel, and based on what we see now, we're excited to do that. So I definitely want to do that. Let mice go out 4 weeks, do a primary tumor surgery, and then maybe separate our groups there based on mets and start treatment at that point. Can we shrink the mets? That would be great.
I was interested in the fact that the TGF-beta decreases the cadherin expression and enhances EMT.
Right.
And TGF-beta, of course, is also an immune inhibitor.
Sure.
Now, in vivo, do you think immune inhibition goes along with EMT?
Whoa. Yeah, definitely. I mean, TGF-beta is is a very complicated molecule. Definitely in that primary tumor, you're going to have a very kind of probably pro-inflammatory microenvironment where there's going to be high levels of TGF-beta. And so is TGF-beta inhibiting an adaptive immune response?
Yeah.
And driving EMT? That's totally possible. Yeah. all I can say to that really is that in that model, we're in a nude mouse, so there are no T cells there, so I really couldn't see an impact on that. But that's a good question. I don't know. That's an important question because I don't know if the EMT we see in the 4T1 model is that driven by TGF-beta. What is it about that microenvironment that's driving that EMT? Because those cells definitely seem to be completely cloaked from the immune system, right? I mean, the fact that we can put 10,000 cells on and get this really robust primary tumor. So is it something about those cells that they're able to exquisitely respond to TGF-beta and allow TGF-beta to do that? So it's not a very good answer. Yes, I think it's the short answer.
So with regards to your data on the localization of FGFR going from the junctional complexes intracellular, can you speculate a bit on whether you think that's just part of the normal recycling process because it is activated, or whether it's sort of being actively recruited into the cell where it's having function? And the second part to that question, because two-part questions are fun, right? Have you looked at the effects of your inhibitor on the localization of the receptor?
Right. So short answer to the second question, no. But I want to. The first one, I don't think it's simply an effect on the localization, because with that short-term TGF-beta treatment, we really don't see any changes in FGFR signaling. So I don't think FGFR is being activated in that context. So I really think it has more to do with somehow that complex becomes broken between ECAD and EGFR. At that kind of time point with TGF-beta, ECAD here is definitely being pulled off the surface. So maybe that liberates FGFR to interact with other proteins. But yeah, that's a good question. We could take those GFP-tagged cells and simply just treat with FGF.
It's interesting to see if then they're retained at the junction.
Yeah, because if you look at those pictures, we actually start to see some FGF in the nucleus. There's definitely there's a large body of literature suggesting that, and how that's taking place, and if EMT has a role in that. One of our reviewers is very interested in that. So maybe we'll All right, thanks a lot.